This morning on BRN, a new study model with clues to anti-aging. Joining me now is Dr. Jiyue of Washington State University. Dr. Jiyue, great to see you. Thanks for joining us on the program this morning.
Thank you for having me here.
Yeah, I'm excited to talk to you. And I love talking about the latest scientific discoveries and research. And you, doctor, you're spending a lot of time researching anti-aging. Can you tell us a little bit about your work and some of the major milestones you've achieved?
I have personally been involved in the aging research or the telomere research for the last 25 years to 30 years. And I started with working as a postdoc under mentorship of two pioneer researchers like Mike Bishop and Elizabeth Blackburn. Both have won Nobel Prize for their field. One is Dr. Bishop is a cancer biologist, and Dr. Blackburn is the telomere pioneer. So over the last 20 years, we have been trying to understand how the telomere and telomere functions, how the enzyme that leads to synthesis of telomere, how it's controlled in our body. And and we're trying to address the question about how telomere is associated with different diseases, and also most importantly, how human age associates with telomere.
So, doctor, I'm a layperson. Obviously, I don't have the benefit of your expertise and experience, but have we been able to figure out, or have you, I should say, and other research been able to figure out how to slow the process of aging? I'm 53. I was telling you, I try to eat right, I try to exercise, but have we been able to figure out how to slow the cellular decay, for lack of a better word?
Well, the short answer is not yet, but we're working on that. So what we know is the telomere, we trying to understand how telomere and is actually shrink, how we can prevent telomere from or slow down the shortening of the telomere, which would be important intervention, trying to discover some intervention that can slow down this process. And the aging is, in a way, is a very scientific challenge as a scientist, but for a lot of people, it's viewed as an unavoidable reality of life. And so I don't think in the short term there will be anything that stop aging, but hopefully we will find some way so that we can slow down a little bit. And most importantly, how to understand this process and improve the quality of life at the later years. And as you know, when we get older, and we can come up with a lot of different health problems. And how do we address these health problems is a key question we are trying to ask in our research.
And doctor, you've done a lot of work with, I guess one of the ways to test theses and to test anti-aging is using mice. You've been successful at looking and using genetically engineered mice in this testing, in this research.
Yeah. Yes. So scientists has been working on this question. There are many scientists in the world, and I would say thousands of scientists in the world, and they study this problem. And in most cases, people have been utilized cultured cells, like in petri dish, and trying to understand how telomere shortens or telomere attrition occur in a petri dish. And this is the problem is that the telomere mechanism itself is not evolutionarily conserved. So, for example, many mammals have a much longer telomere. We humans are born with about like 10 to 15,000 base pair of telomere sequence. but the animals, like for example, mice have 10 times as much of those sequences as we do. And so basically, those animals do not really care about the telomere shortening. They have plenty of this telomere. So the telomere shortening do not affect their aging process. So this allows, although we can really, we can also study what happens in human body, but it's much harder to study in human. And we cannot do, the proposal, some hypothesis and trying to figure out the intervention, which would be much more difficult in human body than if we have a small living organism like mice. Mice are they weigh less than an ounce and they live a little more than 2 years. And so this is much easier the tool to study. But the mice have 10 times as much telomere as we have. So how do we solve this problem? Because what we did is we trying to understand why mice have longer telomere or why we have shorter telomere. And over the years, we have figured out what is the differences, and we use the genetic engineering tools. And based on our knowledge, recently we have succeeded to engineer a mouse with a short telomere, just like a human. And so this is the paper that I recently published in the Nature Communication. And this would be a very nice model for future study of in the aging field.
And Doctor, this data would be available, I believe, or the data from the mice would be available to other researchers. So the plan is, I want to make sure I'm getting this right, that the plan is to take the data gleaned from these genetically engineered mice and share it with others so that it can be used by other researchers in anti-aging.
Yes. So the data itself has already been published in a journal. And we also, the WSU, Washington State University, trying to help to facilitate the distribution of these mice to the different scientists in the world, basically. So this is in progress. So we not only will share the data, but also share the mice itself.
Yeah, I mean, look, I think a lot of us are counting on you and your colleagues, Dr. Jiyue. I mean, I don't want to live forever, but I think it's important for longevity, but also you want to minimize the disease, right? I mean, you want to have a good quality of life. You want to eliminate the possibility of disease. I mean, that's what tends to happen at the end of life, is that things start to break down. Not only do you lose your balance, but you start to get diseases like cancer, et cetera.
Right. Well, think about this. 5 leading causes of death are cancer, lung diseases, kidney diseases, and heart diseases and stroke. These are the leading causes of human fatality at the older age. And but all these have been shown. There are scientific data showing that these diseases are tightly correlated with telomere.
Yeah. Well, it's excellent research, and I hope, selfishly, I hope I'm around when you guys, when you have a medical breakthrough, because my wife and I are counting on you. Dr. Jiyue, we're going to have to leave it there. Continue the great research. It's great to talk to you, and we look forward to having you back on the program again very soon.
Thank you. It's my pleasure.
And don't forget to subscribe to our daily newsletter, The Morning Pulse, and support all great BRN programming. Details, of course, at our website. And we're back again tomorrow for another edition of BRN. Until then, I'm Jeff Snyder. Stay safe, keep on saving, and don't forget, roll with the changes.